Okay, this is a little video to help you through genetics questions. This particular question is on page 7 of your task booklet on genetics, your inheritance one. Practice what you need to do. So the first thing to do is to read the question and annotate it as you go along. So here we go. Black fur in rabbits is determined by a gene big B, which is dominant to brown fur. So any rabbit with black fur could have a genotype big B, both coding for black fur, or big B, little b, where the dominant allele will sort of override the recessive allele for brown fur and still give us a black furred rabbit. And we've got brown fur, where the genotype of any rabbit with brown fur will have to be little b. We've also got, it says the gene for normal length fur is dominant to that for short fur. So the same principles apply. Normal length fur can be big N, or big N, little n. And short furred rabbits are all going to have to be little n. The other thing to notice is we're looking at two completely different characteristics. We're looking at fur colour, black or brown, or how long the fur is. So this is turning out to be a dihybrid cross. So we're going to need to deal with two sets of alleles. So in the first cross, a homozygous black rabbit with normal fur. So homozygous, both alleles are the same. Our rabbit is black, so it's got to have this big B allele. Black rabbit, big B, normal fur, normal length is dominant, big N. Was mated with a homozygous brown rabbit with short fur. So brown, little b, and fur, short fur is little n. In the second cross, one of the offspring from the first cross was mated with another homozygous brown rabbit with short fur. So we need one of the offspring, which we've not worked out yet, was mated with another homozygous brown rabbit with short fur. So that's again little b, little n. With suitable genetic diagrams, show the results of these matings, and we've got 7 marks. So if we look at the side headings, that might tell us where those marks are. So we've got parental genotypes, possible gametes. We've got a little space to give our first generation and what sort they are and how many there are. Perhaps their phenotype and their genotype, 1, 2, 3. Parental genotypes, 4. Possible gametes, 5. Little space to do that Punnett square. 6. Offspring genotypes, offspring phenotypes, 7. So we need to write something correct next to each one of those. So parental genotypes, the thing to remember, we're dealing with a dihybrid cross, so we'll need 2 letters, because in the parental genotype they've got their chromosomes in pairs, and we're going to need 2 letters for fur colour and 2 letters for how long the fur is. So we worked out here that we've got homozygous black rabbit, both alleles are the same, with a normal fur, homozygous, both alleles the same, mated with, so put our little cross sign in so we don't get confused, homozygous brown rabbit, little b, short fur, little n. So possible gametes. Our first one has got to pass on a big B allele, and that could combine with big N. So that's gamete type 1, or we're circling gametes so we don't get confused. Big B could combine with big N, it's the same. Big B with big N, it's the same. Big B with big N, it's the same. We don't need to write it down again then. Crossed with our other rabbit, little b can combine with little n, circle it. Little b with little n, little b with little n, little b with little n, they're all the same. This rabbit can only pass on big B alleles and big N alleles. This one can only pass on little b and little n. So in our F1, remember we put one parent's gametes here and the other one there, merging them together, keeping the letters together and capital first so we don't get confused. Big B, little b, big N, little n. So all of our first generation, all of those rabbits are big B, little b, big N, little n. Black fur because it's dominant, normal length. So our second cross, go back to the question. In the second cross, one of the offspring, that's this guy, from the first cross mated with another homozygous brown rabbit with short fur. This is our offspring, parental genotype, big B, big N, little n, crossed with homozygous brown rabbit, homozygous, little b for brown, short fur, little n, little n. So our possible gametes. Big B can combine with big N and give it a circle. Big B can combine with little n. Different, so we'll have to write that one down. Little b can combine with big N. That's different, we'll write that one down. And lastly, little b can combine with little n, and that's a new gamete type again, crossed with little b, little n, or little b, little n, that's the same, little b, little n, that's the same, little b, little n, that's the same. Our Punnett square then, I'm going to put my 4 different gametes along the top and my 1 gamete type from the other parent down the side. You just transfer those in. And here we go. Big B, big N, little n. Offspring genotype. Big B, big N, little n. So this rabbit, big B is black and it's got normal length fur. Stick a comma in. Big B, little n, little n. Big B, little n, little n. And this one has black fur again, but it's short. I'm going to make that a semicolon to make sure we know. We've then got little b, big N, little n. So this rabbit is brown and it's got normal length fur. Now I seem to have run out of space and I don't want to put it where the examiner is going to put their ticks, so I'm going to put the next one underneath. So I've got little b, little n, little n, little b, little n, little n, and that one is brown with short fur. And when the examiner reads this, they're going to read that. But I want to be sure they know that I've matched it up. So I'm just going to put 1, 1, 2, 2, 3, 3, 4, 4, so that they know that I know which rabbit type belongs to which genotype. So turning over. Himalayan rabbits, never heard of them. Belong to a variety in which all of the individuals carry the dominant black gene. So these are black rabbits. Okay. However, but it doesn't tell us whether they're big B or big B, little b, but they've all got the dominant gene. However, the fur of newborn Himalayan rabbits is white. Weird. As the rabbits grow older, the ears, nose, paws and tail grow black fur. Interesting. When a patch of white fur was shaved from the back of a rabbit, which, what bizarre thing to do, okay, and kept in a cool environment, let's stick it in the fridge, why not? The fur that grew back was black, so cold equals black. The normal body temperature of a rabbit's 33 degrees centigrade. That's quite warm, a bit like us. So, the presence of the big B gene in the nucleus of the rabbit cells controls the reactions. So we're looking at the nucleus, so we're talking the DNA there. Controls the reactions which produce the black pigment. So we've got a DNA code, black pigment. In the chain below, insert the processes or terms that fill the gaps linking the gene to the production of pigment. So genes, we're looking at a section of DNA that codes for a protein. So let's have a look. So we've got gene, something, something, protein synthesis, something, pigment synthesis. So we've got 3 gaps to fill in. So between a gene and making a protein, we've got transcription first, that's making the mRNA, and then we've got translation, so that's quite straightforward. But what could be between making the protein and making a pigment? That's to do with something to do with temperature. Well, my guess would be that we are making an enzyme. So somewhere on the ribosomes we're making this sort of polypeptide chain and we're modifying it to make an enzyme. And I'm going to put in synthesis there. Explain the experimental evidence for the effect of temperature on the process. So if we're talking about enzyme synthesis and the cold colour is making it black, then in some way when it's warm it's going to be not black. So we're not making the pigment when it's warm. So that's not when warm. Which I suppose, if you think about the ears, nose, paws and tails, they're kind of at the extremities of the animal, and that might be cooler. So let's have a look. So what we're saying is that the black pigment is only synthesized when temperature is low. Now why could that be? I don't think it's being denatured particularly, because we're only dealing with temperatures of 33 degrees centigrade still giving us white fur, so it must be, the enzyme must be inactivated. Plus I think if it was denatured, when you shave your rabbit when you shave your rabbit and stick it in the fridge, it would still grow back white fur if the enzyme had been denatured. So we better use the word inactivated, I think, rather than denatured. So, sorry, I'm using rabbits in fridges. Suggest explanations for all the newborn rabbits having white fur. So, being a mammal, this is going to have internal gestation, and so they have been, prior to birth, in a very warm environment where we've inactivated that enzyme. So prior to birth, we use some biological words here. The rabbits were in the uterus and therefore warm, 33 degrees, we've been told that. So the enzyme is inactive. And no black pigment is produced. The colour pattern in the adults then, so these are white rabbits with little black noses, ears, paws and tails. So the colour pattern in the adults, we can say the black pigment is made at the extremities where the temperature is cooler. So we'll put lower temperature, you don't say cooler temperature, do you? Okay, that's it. Good luck. Still don't know how to switch it off. I'm just going to have to put the doodah off. Oh, I stopped it. Oh no.